Hello and welcome to another episode of Drug Discovery with IronCure. I have with me Dr. Tejas Gupte, who's a drug discovery expert at IronCure. I'm Sukant Khurana, founder of IronCure. So we are here discussing why drug discovery is damn difficult, how IronCure is there to make it somewhat easier, what our objective is in terms of using artificial intelligence, computational approaches, using knowledge from natural sources, basically getting inspiration from wherever we can to make drug discovery a faster and a better process. We really hope that medicines don't cost people as much and they don't take forever to come out in market. That's our agenda. So we started our podcast with a discussion where in the last episode, Tejas told us one of the reasons why drug discovery takes very long time. And what he had said, I'll just try to summarize in a line or two. He had told that there is a lot of biomedical data, but it's not connected. So to synthesize it, be able to use machine learning, that data doesn't exist. And I was saying how we, as IronCure, are using the power of citizen science to try to build those data sets, try to actually make sense of almost all the biological data, which is a massive task. I mean, people will laugh at us if we say we are trying to make sense of all biomedical literature, but we are trying to do that. So that's the aim, and let's see how far we go with that. But today we'll discuss other issues why drug discovery is long. So Tejas, entertain me. So when we hear these reports of cancer cured, and we later realize that somebody worked on 2 cell lines and found some compounds which killed cancer cells. Sometimes you even find they kill cancer cells, and you don't have even a control of normal cells. So maybe they're killing normal cells also. But sometimes you get cure of cancer in mice and you get cure of some HIV in some other animal. Why does the drug not come out to market? Why do we have so many reports of miracle cures and very few medicines?
Sure. That is a loaded question. So let me break it down, right? So first is there are reports about drugs which have activity. Why are these drugs not being brought out into the market? When we say a drug has activity on a cell line, that is just pure pharmacology. In a good report or a good paper, good manuscript would essentially start out by saying, this is the cell line, this is a cancerous cell line. The origin of, first and foremost, we should know what is the origin of that cancerous cell line, right? What cancer in the body is it mimicking? Is it mimicking any, or is it just an immortalized cancerous cell line whose properties have changed tremendously over the passages that it has gone through? That's a very basic information that we must have. Okay, so let's assume we have that information. Then what the scientist is doing in a good study is basically dissolving these test compounds in either water or DMSO, the two most commonly used solvents, or ethanol, and throwing them at increasing concentrations on a series of cells coming from the same starter set. And then you're asking, at what concentration of the drug are these cancer cells dying? At what concentration are 10% dying? At what concentration are 50% dying? At what concentration are 90% dying, right? And in how much time are they dying? So these are so concentration and time, these become the two parameters here.
Absolutely.
That is the very basic pharmacology that we are doing. Now, from this, all that you know is that one out of some of the drugs that were tested is probably killing these cancerous cells, right? So now, as a bedside scientist, right, my question is, did they have the appropriate controls? Did they have positive controls? Did they have negative controls? Were their cells cultured under proper conditions? Are they dying because they put too much ethanol as the control vehicle? Are they dying because they put too much DMSO as the control vehicle? These are precautions that every good scientist has to take, must take, and good papers, they do take these precautions. So let's assume these precautions were also taken in the paper. And so you can believe now that this drug does indeed kill this particular set of cancer cells. Now, between the identification of this molecule and a patient actually being allowed to consume it, there are a huge number of steps, right? So the steps are, is it applicable to an actual cancer? Because as we said, this could be 1 or 2 cancer cell lines which are related to a certain kind of cancer in the body. But does that also then work on the cancer in the body? What is the toxicity of this compound to the human being if they consume it, right? So that is called the pharmacodynamic effect. The pharmacokinetic effect is what is the effect of the body on the compound when it is consumed. So how does the body absorb it, distribute it, metabolize it, excrete it, right? These are studies that need to be done. Then we also need to know, if such a compound is consumed, is it only going to affect the specific protein or mechanism which is involved in the cancer, or is that just one of the many possible targets that this drug molecule, so-called drug molecule, has, right? And then what could be the possible side effects? Because what you want is, you want to minimize side effects. You want to increase the effect of killing the cancer through a very specific mechanism. So there are so many steps, and each of them takes time. Now, the reason why drug development used to take such a long time previously is that many of these steps had to be done experimentally. And I think over the last 30 odd years, a lot of these steps can be done in silico to some level or the other. You know, you can't say all of this can be done in silico, but a lot of it can. There are good models to predict a lot of these mechanisms and effects in silico, which has brought down the cost and the time to a certain extent. But there is no substitute for doing some of these studies in biological systems. Then again, there's the difference in the metabolism between small mammals, human beings. Small differences between the protein sequences of different mammals itself, which sometimes, you know, a molecule can look like a drug when it's put in a rat, but may not act as a drug in the human being, or vice versa. It may actually be a drug in a human being, but not act as a drug in a smaller mammal. So there are just so many complications here that you have to be very careful. Now, another thing that I think modern drug discovery is doing very well is, we realize that affecting the active site of a protein or enzyme is just one of the ways of targeting it, right? And previously, we always looked at more water-soluble compounds because the active site is exposed to the solvent. But what we have realized now is that the protein-protein interaction actually gives specificity to a pathway or to a phenotype. And many protein-protein interaction interfaces have hydrophobic hotspots, which means drug molecules which were earlier discarded for being too hydrophobic or for being, you know, amphiphilic, can now actually be included as candidates because they can affect these protein-protein interaction interfaces. So there is a lot of you know, movement going forward in multiple directions, both in silico and in the wet lab. And because there are all these multiple steps between the identification of an effect and its actual ability to be consumed and show the effect on a human being, that is where it takes a lot of time.
So I am really glad you mentioned other thing, which actually a lot of AI companies, such as us, tend to ignore. We are not just an AI company, we are a drug discovery company using AI, and we're using AI for certain other healthcare things too. But people tend to ignore the fact that better experimental preparation, improved ways of delivery. So there are other innovations which are happening. But
absolutely,
as you might be put, assuming it's not a farcical pharmacological report. There are a lot of steps which go from pharmacology to final drug going out in market. And after drug discovery, there is drug development thing. In terms of clinical trials, takes a very long time.
absolutely.
That's when you hear that somebody says that in 2 months they're going to launch a medicine. You can pretty much be sure that it's a joke. During COVID-19, we are seeing a lot of such jokes, but
yeah, we seem to be living a pandemic of jokes as well right now, unfortunately.
Yeah. So coming to this, it was a great learning experience, Tejas. Thanks for your time. I would like to say that IonCure as a company is always welcoming of citizen scientists. Our website is ioncurerx.com. ION as in iron, CURE incurerx.com. Please visit our website. Feel free to join us. We will be very happy to see if there is a good fit related to data science, related to drug discovery, related to artificial intelligence. We apply fourth industrial revolution also to technologies, to several other things. Get in touch with us. And if there is common interest, we'll do things together. And we hope that in addition to people who are scientists, other people are also listening to this, who are common people who are hoping to find out how drugs are discovered. They should keep looking at what we are doing. Hopefully, we'll contribute to more and more medicines for various diseases in the times to come. Thank you very much for your time.
Thank you.